Thanks so much. Really excited to be here. It was wonderful being at the conference last year too. So Bridget, who's over there in the back, she was the one actually doing all the sampling of the AP heroes and the associated controls. And I was there briefly. I was in the middle of vacation, but actually wasn't that far away, so I popped in for a day. It was really a wonderful conference, really impressive what you've been able to do with this really tough disorder where it's not monogenic, right? So that does pose certain constraints on what you can do. So Frederick Vigneault, who's our Chief Scientific Officer, he's also on the call. I think he's in the lab right now. And we have Rahul, who heads the computational team, Michaela, who is Business Development Associate, more on the rare shift side, and Erica, who leads all things tadpole in the lab. So that's the Unravel side here. And there's a few more people. And today we had a bunch of interns start, so it's a little bit of a beehive back there. Yeah, let me share slides and, oh, I usually need screen sharing. Can you guys see it okay?
It is in the presenter mode, so if you wanted to flip it so that we see the slides and you see the presenter mode, or this works as well.
Okay, let's see. All right, is that better?
Yes, thank you.
Great. Yeah, so again, the idea is to share a little bit about how Unravel works. It is a very different process, and so there's a little bit of an intro to how we work. And Lauren mentioned nasal swabs, and that always gets people scratching their heads. So we'll discuss a little bit how that works. But the whole idea is that it's a personalized medicine first approach that we take in general. And it's really well suited for AP heroes where, given where the chromosomal breaks and modifications happen, they're all kind of unique, right? And we see that from the phenotypes of the patients. And so the whole idea is this personalized medicine approach can give you a much clearer insight into the very complex biology. And so before I dig into all of that, just a huge thank you to the study participants, both the patients as well as the families who were so instrumental. Again, this is what enables research into the chromosome AP disorders, and actually let us work on developing treatments. And again, huge thanks to Project AP's funding that funded this whole study. So just taking a step back in terms of typical drug, target-based drug development that I think everybody on this call is familiar with. You start with a target, then there's some preclinical development, then clinical, and then you hope that a drug can actually make it to the market. And this is really challenging for rare disease, for poorly understood diseases, and for very heterogeneous diseases. And the biggest challenge is the clinical one. So many drugs that start early on, go through preclinical development, they go into trials, and the vast majority of new targets fail in the clinic, mostly because they just don't work, not because of toxicity, not because of anything else. And so this process is really where pharma has not succeeded very well in terms of rare and ultra-rare diseases especially. And so why? And so we thought about this as we were starting Unravel, and our whole thought was, well, we're targeting diseases in this traditional drug development path, but diseases are not patients. And so on the right, you have some great examples of diseases, these kind of general descriptions of Angelman, lipedema. We work on various diseases in this group, but they're not patients, right? And so the corollary of that is that patients, not diseases, actually respond to treatment. And again, this is not a new concept. This goes back to the early 1800s, but it's about those individual patients. You can see actually a few in our office here, where that we work with, that are, that's actually the problem that you're trying to solve, right? It's not this amorphous disease. It is patient 1, patient 2, patient 3, little Bobby, little Susie, not, and that's the problem that keeps us up at night. And so what Unravel has done is really doing drug development driven by patients. And it's the same little logos that you saw earlier, but it really starts with patients. The whole idea is let's start with the problem that the patient has, not some amorphous idea of what the target should be or could be. We can get there, but let's start with patient data. And that's really what informs even this study. And going a little bit deeper in terms of how we work, the kind of underlying concept is that a patient, it's not just the mutation, right? So often we use this shorthand for, oh, it's a 8P patient or whatever, summary of their mutation. Well, the mutation doesn't necessarily define the patient. It's the genetics, including the mutation, but not limited to that. There's 25,000 other genes and non-coding regions as well. The environment, medication, physical therapy, sleep, diet, all that stuff matters. And then time. Patients change. The DNA does not. The mutation does not in these cases, right? And yet the patient is changing. And so how can we capture that? So we use RNA and use the RNA profile as a way to complement the more commonly used DNA analyses because it's dynamic. And again, all the kind of environmental parameters I mentioned, those play a role. And so then this enables our model of human health to really get a better understanding of the underlying biology and the dynamic aspects of it. And then one insight that we had in this process was that these complex biological networks, rather than the individual genes, are what drive clinical function. And so if we want to look at patient outcomes, whether it's just the progression of the disease or response to treatment, we really need to be looking at this from a much more holistic systems level perspective than the gene by gene approach. And so if I just give you an example of a typical healthy network, completely made up, it might look something like this. Think different genes talk to other genes. And again, it's not necessarily, certainly not the RNA, but it could be a metabolite of one enzyme interacts with the protein, let's say like a transcription factor, which can then change the gene expression profile of something else. But it's a network, right? They're somehow interacting. And then you have a mutation. And then this messes up all these nodes around that, what it's interacting with. And then those perturbations propagate, and you have some sort of unhealthy network. And then what we as Unravel are trying to do is look at all the ways you could solve this. And one way, yeah, you can target that mutated node. Now, in chromosome 8P, that's a challenge. It's not one gene, right? What can we do? Well, can we rebalance these networks in some way, right? And the analogy that I like to use is that it's like Google Maps, but for health. You have a starting point, which is the patient's network, and you have a destination, which is, let's say, the control network. How do we get there? And we don't really care how, we just care that we get there, that we can somehow rebalance the network. And so what we're able to do is forecast how patients respond to therapy. So we use patient RNA signatures together with drug RNA signatures now, because we want to understand what a molecule actually does, not what it was developed for. And together, using our AI network analysis, we can ask several questions. So we can ask, will it help this patient? That's sort of the personalized medicine angle. Can we treat all the patients? The answer may be no, but there are subgroups that are similar, and we can treat those subgroups. Are there better treatments, right? Are there some better or worse treatments within the same drug class? Are there better treatments to what's already used in standard of care? There are all these different flavors of that question. And then the opposite, is there anything that would actually hurt a patient? Even though it might make sense, would you actually predict the outcome to be negative overall? And so with that in mind, we started this Project 8P pilot. And again, this was the population study, is 19 patients. That was all done at the conference last year, at the symposium. And then there was another personalized study completed. So it's 8 samples over 48 hours instead of a single sample at the conference. And I'll share both results. And there are more patients in progress as well. And then all that data is shared back to the foundation. And so the idea is that researchers can have access to this data. And the way it was done was really you have a patient, male or female, and then the same sex matched first degree relative. So in this study, it was all parents that were the controls. And there's several goals here. So one goal is, are there potential treatments that could be used to help patients and inform clinical care, right? So that comes down to, well, it's in a very heterogeneous population. Are there subgroups that might be more similar to each other from a drug response standpoint? And can we find those drugs that could actually help them? And then from that, use that to understand the mechanisms driving the disease and other aspects, but it's really putting the patient and drug response first, right? How can we get to a treatment? Everything else is secondary. And then this really is a first step to understand the patient population. So looking at, how do we mitigate adverse effects? So in clinical trials, you might get non-responders and they have all the side effects and none of the benefits. How can we get rid of that as much as possible? And then what would be the strategy of getting to a clinic? So we're about to start running 2 clinical trials, and we put a lot of thought into what does the clinical trial look like, and then work backwards from there. And what data would we need? How would we think about patient inclusion, exclusion, and other aspects? And so the idea is that this is a pilot. This is by no means an end-all, be-all completed study, especially given the very heterogeneous nature of this disease, but it's a place to start. And all those questions that I showed earlier, you really can't get that from an animal model, and there isn't one for AP. But even if you did, you can't answer those questions with models alone. And so the way this worked is that we normally we swab anywhere in the world, and this was primarily at this conference, plus at least one patient who was actually ex-US. Mostly it's done through the mail, but the whole idea is that we can basically get genetic insights into the patient's condition, again using RNA, run it through our computational platform, which is called BioNav, and then it gives you several outputs. So it looks at what's going on with the condition, especially over time. We can resample patients. We can find new approaches to treating the condition, predicting new or repurposed therapeutics, and kind of coming up with essentially a treatment strategy as well at a population level. And so oftentimes a question that we get is, why the nose? How is it relevant to neurological disorder, et cetera? Couple answers there. One is with neuro disorders, you're always dealing with surrogate tissues, especially for controls. People don't like brain biopsies. The other part is that we actually collect a very large number of coding and non-coding transcripts out of the nose. And so far, I think in every single case where we've been asked, would you be able to see our gene? It's strictly neuronal, we see it. The idea is it's a kind of a newer tissue that people haven't focused on until this lovely pandemic. And then the other part is it actually does have a very strong representation of neuronal genes, even though it's not this neuronal tissue, but it is very complex. It's not skin. And we get a very good representation of a lot of neuronal pathways. And so taken together, one, you're always dealing with a surrogate. This is a pretty good surrogate. Better than blood, I would say, but we're going to make that comparison. And with the same patients, actually, because those samples were collected at the same time. And it lets you also let patients self-collect, or the caregivers self-collect, and ship this all around the world. So actually, it's a very, very user-friendly, very practical source of data, and it seems to be good. And so now getting at the results, so this is a principal component analysis of the gene expression patterns. And so the patients are in orange and the controls are in blue. And you can see overall there isn't very good separation, right? Not too surprising for such a heterogeneous disorder. This is just based on gene expression. And this covers a whole range of patient ages, so 0 to 18, actually a number of zeros and a number of kind of late teen patients. About evenly split, male, female. And then it was, in all cases, it was the parent. So these were all, kind of roughly 25, 30 or older controls. And what's interesting is that even though there isn't an overall separation, which we sometimes see, but it's typically not as strong at a whole population level, what we see here, and this is actually, shout out to Lauren for proposing this, if we compare sort of that direction between the blue and the orange paired dots, so if we just look at these two where they're very nicely separated out, what we see, I think in every single case, is that the patient is always to the left of its corresponding control. And so this PC1 axis, which is a combination of a whole bunch of gene expression weights, pretty consistently distinguishes who's going to be the patient versus the control. So there's something there at the gene expression level. That tends to be not where we put the focus, because again, what I mentioned before, it's really that network interaction that drives the drug response. And so what we do is we take that gene expression data that is represented in that PCA plot, and we run it through BioNev. And so we basically predict drugs for each individual patient relative to their control. And then we put it in the context of the population. And so the UBIDs are in individual patients or patient control pairs. And then we rank the drugs from basically, based on various scoring that we do, and happy to discuss that in more detail. And then we basically rank them from most therapeutic to least therapeutic, where least therapeutic is actually harmful potentially. And we then can correlate the drug names with their ranks across each of the pairs of patient groups or individual patients, right? And the red areas are positively correlated, such as here. The blue areas are negatively correlated, so that means that the drug that's over the whole list of drugs, the top drugs for, let's say, patient UB117 are actually at the bottom of the list for UB123, as an example. And then we can cluster patients based on the similarity of these overall drug responses, right? So this is not any one drug, really looking at these drug networks overall. And one thing that we started seeing is we essentially annotated it, a couple different computational methods you can use, but basically there are 4 subgroups if you do this analysis. And so that's shown in this left column here with the 4 different colors. So this was added by us. You can see the sex, male, female, not a whole lot of clustering. There's some little subclusters, but it doesn't separate out these other patients. And then this data set was mostly the individual patients with 3 deletions and 1 unknown. But there's no clear clustering there either. Removing those doesn't really change the clustering pattern. It just removed those out of the groups. Here, we still have these same 2 corner groups that are the clearest groups, and then potentially, again, kind of 1, maybe 2 intermediate groups, but didn't substantially change the structure of the population. And so now if we look at, so this is the drug list overall, so it's several hundred drugs, right? So that's how this is compared. If we now look at, okay, well, what's actually driving this? And if we look at the individual drugs, and just as a general policy for both IP, but also just liability reasons, we just don't share the names of the drugs in public presentations, but of course the foundation has all this information. So then we work with foundations to basically figure out how to best present the actual drug names when it's appropriate. But basically, these are all individual molecules. They're not necessarily drugs per se. They could be, other compounds, all purchasable compounds. None of these are new. We can start looking at the scores, right? And so like the red coloring shows the highest scores, and this is 0 to 100, and then blue shows the lowest scores. These are top ranked overall, and we combine both the mean as well as the standard deviation, because what we want is the most consistently high ranked drugs. And so the standard deviation gives you the consistency, and then the mean gives you obviously the mean. And what's really interesting, and Lauren actually pointed this out earlier, you very quickly get to basically the standard deviation driving the scoring rather than the mean, and that's shown here with these kind of mid-level drugs that are kind of blah drugs overall, but they're very consistently blah. And the fact that they show up this high basically says that there's a need for further stratification. You can't really draw a whole lot of conclusions from 8P as a single population. And this is not surprising given what we know about 8P. And so there are a couple standard stratifications that we do anyway, and then I'll talk about what was more the data-driven stratification. So one very obvious one is, okay, does the sex matter, right? We do have these sex match controls. It was, I think, 8 versus 11, so pretty well-powered, relatively well-powered for the population size here. Are there any differences? And there were some slight differences. It wasn't huge. There were definitely more metabolic modulators, so kind of your diabetes drugs and statins, I think, a few other ones. For female patients, very much pronounced anti-inflammatories for the male patients, but overall that wasn't a big discriminating factor, but there were some differences there. But then we wanted to start seeing, okay, what about the deletion versus the inversion duplication deletion? Interesting, if you just ignore the table for a second, there was a negative correlation, which is not too surprising, because then with the duplication aspect, you you have more of those genes being expressed potentially than in Dup-Del heroes than in the deletion alone. And the correlation coefficient here was negative 0.28. So that's actually pretty negative. We've seen more negative, but that's actually pretty negative. The only limiting factor here, just to note, is this is based off of 3 deletion patients, and then 15. We excluded the unknown one, but so out of those 18, there were the 15 of the rest. But that said, there were actually some drugs that were shared for both. And so this is basically looking at the best scores overall between these two groups. There's no obvious clear pathway, but there were some hormonal differences, some hormonal drivers, histamine, there's some inflammatory components. There might be something there, but overall, again, it goes to the point where this other stratification is more necessary. So now what we have here is, again, looking at the inversion, duplication, deletion versus the deletion. This is looking at which drugs are best for each of these groups separately. And all of a sudden, the deletion you can start seeing how very different it is. And so basically it's saying the drug 1 is actually really highly ranked if we take all the inversion, duplication, deletion patients together. And when we look at these individual subgroups, that's actually looking, there's enough drugs here that are looking pretty promising. All of a sudden, when you start doing the stratification, those kind of 50th percentile, as they call them, the blah drugs before, those go away, right? So you start getting a little bit more clarity in terms of what might be happening. And now we can also start looking at going back to the gene expression profiling, and looking at some of the genes. And this gene list came from the Project AP Foundation. My sort of top genes of interest. There's obviously a lot more genes than this that we looked at, and I'll show you some plots in the next few slides. But these are some of the genes that these top 4 were significant, and then these other ones were not significant but still a little bit perturbed. And so if we start looking at, how do all the genes across the 8p arm behave? So this is color-coded by the chromosomal subregions. Here's just AP22, just to very quickly orient you. And this is looking overall across all the whatever 18, 19 heroes versus their respective controls. So it's a paired comparison with that red line being sort of the 100th percentile, right? So what's the sort of average for the population? And you start seeing, an increase over here, a little bit of an increase over here, and then some decreases and even dropouts. So these genes were just not detected, so that would be coinciding with a deletion or a very, very lowly transcribed gene. If we look at, again, a subset of these were, again, of the genes that were more of interest, you see a stronger pattern here with just those genes, kind of an underexpression here and an overexpression here. And this is on a log scale, I should mention. And then if we plot it, going at the individual patients, if we look at this individually, we have, again, the subgroups here, and this is only looking at gene expression. So the subgroups are from the drug predictions. We have the sex of the patient, and then we have the mutation part here. You can start seeing some interesting clusters emerge. So basically, this kind of 8p22 part of the chromosome is overexpressed. And we see that here with this group of patients as well. And this does cluster a little bit with the drug subgroups. So there's definitely some mechanisms that are now we're starting to correlate a little bit with the gene expression, but again, not of any one gene, but of this collection of genes with the drug response profiles. And what's interesting is if we look at just the 3 deletions, you can see this one just doesn't really have a whole lot. If anything, it's kind of on the low end. This is a linear scale, and I'll show a log scale in a few slides that captures the negative part much better. And you see that here's another deletion, right? So you don't see this overexpression in the deletion patients. And again, here you see maybe a slight increase, but overall it's much lower than everybody else. And then this unknown, to me, looks like it could be one of these sort of low expressing duplications in Dup-Del patients, or it could be a deletion. And maybe that's why the patient's undiagnosed. There may be some other complicating aspects to this, or it's not clear which one they are. But one thing that we had, right, so the limitation of these population studies is you have one sample. So you can't really do a lot of statistical analysis on any one patient. And at a population level, you have heterogeneity. Where the breaks happen, it's not exactly consistent. And so we have one AP hero that it has an inversion duplication deletion. And in their registration for the study, they noted these genes as sort of the diagnostic findings. We're trying to figure out exactly what they had in their report, but what's shown in red corresponds with this region right here that we see increased. And what's really interesting with this patient, you start really seeing these deletions, or what looks to me like a deletion where really a lot of genes are dropping out or very significantly decreased. Whereas this part of the chromosome looks pretty consistent, or much more consistent than the left side. Again, looking at more just the genes of interest, again, this came from the foundation. Now those genes from that patient, again, that were reported are in red, and most of those are in the overexpressed category. And again, this is a log scale, right? So this is, these are probably 12, 15 times more expressed. And just to give us a better perspective on what might be happening at the chromosome level. And so going back to that same plot that I showed earlier, that heat map, again, you see this similar pattern where, especially in the 822 region, all of these are individual samples at different times of day. They are overexpressed here, and then you have these sort of lower expressed regions as well. But this banding pattern, what's interesting, it persists even into the population. And so, again, this is the data that I just showed you. This is the UB194 was the personalized study. This is the data I just showed you on the previous slide. Now in the log scale, so you can see the negatively expressed or underexpressed genes much more clearly. And you can see this lines up really well with this subgroup here, really, really nicely. The other part that you see even more so is that if we look at the drug response subgroups, the purple and red subgroups are effectively clustered together here. And the blue and yellow subgroups 1 and 2 are more clustered here. And it makes sense, right? You essentially have the patients with this very high overexpression in that region of the chromosome and ones that have much less. Still overexpressed, but much less. So you start seeing these really interesting patterns, but we can now go back and forth between gene expression level analyses, which are more the traditional ones, and linking that to, well, how do we actually predict a drug response? And that's what's really coming out of the BioNav study here. And so just to summarize, again, this is just kind of the beginning of this collaboration with Project AP. There's a lot more that will be happening with other samples, but what we have done so far is we analyzed the AP hero population, again with RNA-seq using nasal swabs for this particular study that's showed here. We identified up to 4 subgroups, maybe 3, but definitely not less than 3. And the gene expression changes align reasonably well with the chromosomal position, but even at the gene expression level, there are subgroups. It's not, all the patients are not the same just because they have the INF2del type of mutation. And then the personalized study, it does give you far more statistical confidence in the results because again, it's resampling the same 2 individuals over time. So there's no inter-subject differences. And then the personalized study does agree with a subset of the population results. So again, kind of reinforcing both data sets. And then what we're working on now, we have, of course, a list of drugs. Typically, we would say, let's either we use Xenopus laevis, make tadpole models, go to mice, or whatever. For AP, that's not really a possibility, or not an easy possibility, certainly. But there are various lines, of course, that the foundation is supporting. And then what we do not advocate this, but what does sometimes happen is that you could, in theory, go straight to, let's say, an observational study. Some of the drugs are, by all accounts, quite safe, or even supplements or something like that. So there are all these different ways that could be used to move on to next steps, but this is kind of where we're at right now. Yeah, so with that, thank you guys for letting me present.
Thank you, Richard and team. We're so grateful for your time and for sharing all of this with us. I have so many questions that I am so eager to ask, but I am lucky enough to get time with Richard and his team outside of this. So I would love if there are any questions from our group here. Please don't hesitate to ask. You're welcome to unmute yourself or put the question in the chat, and I'm more than happy to help read it as well.
Can I ask for a clarification? The groups you identified, was that based on their responsive, the comparison with how the drugs would change their expression profile, or was it based on their expression profiles only?
So which ones?
When you say you identified 3 to 4 subgroups, I'm trying to understand how were they grouped based on their how their gene expression was the similar or different, or was it also about how the something to do with the drugs? I'm trying to understand what the subgroups and how you identified them.
Yeah, those are all with looking at basically simulating the effects of about 40,000 molecules on each patient and looking at how similar are the responses on a patient-by-patient level, and then putting them into groups based on that.
Okay, so it is responses to the drug.
Yep, yeah. So the groups 1, 2, 3, 4 are always the drug responses. And then we did cluster kind of towards the end, looking at clustering by gene expression. But the annotations of the groups were still based on the based on the drug predictions.
Right, yeah.
Right, so the idea is to
And you
link
were
them.
seeing some overlap between the clustering based on the
That's right.
gene expression and the responsiveness to the drugs. Okay.
That's right.
So my second question is, I mean, I guess the assumption is if there's something abnormal that's affecting the network, that if you make it more towards normal, you're going to have an effect on the disease state. But you don't really, if you don't know exactly how that abnormality is contributing to the disease state, you don't really know how much of effect you're going to have by fixing it. Right? You know, it could just be, you know, a minor component, and so you make it normal, but it's not doing enough of the underlying disease state to really see any clinical benefit.
Right, yeah, so the way we score the drugs is on kind of looking at the network in several different levels. So one is, we do include, well, how similar is the just gene expression overall being restored back to normal? Looking at just gene by gene, kind of like a barcode, you know, what happens? So that's one. The stronger data comes from kind of networks of genes, right? So again, the idea is to go from just single genes all the way to these whole network effects, and then each one has a score, so 3 different scores. And then the overall drug rankings are based on a combination of those. So we do, based on how those score, we do get a sense of how extensive the effect is. Now, to your point, you're right in that, do we know it's going to help with this symptom or that symptom? No, we don't, right? But what we have consistently seen across many programs is the broader, the more consistently it fixes the network overall, the more symptoms get targeted by it, right? So we don't know which one with any one drug before we test it. This is where, for example, testing in animals is really useful, because does it help with seizures? Does it help with motor function, right? And let's say the patient has both. Well, you don't know which one necessarily would help, but you try it and see, and then that could help down select it.
Okay,
Yeah, but hopefully that helps clarify it.
thank you.
For any of you that missed some of the earlier parts of the presentation, then feel free to ask away. Thank you to the Unravel team, of course. I guess I don't know if Lauren, you were gonna ask this too, but I was just thinking, like, Laura, based on some of your questions too, like, would it help if you heard 1 or 2, you know, case stories essentially on where maybe some of these drugs were tried and how they arrived to it? Does that help you? I mean, it won't be an 8P obviously, but with other rare diseases? Laura, does that help?
Oh, sorry, that question was
I
to,
know
that
that- I
question
know-
was to
okay,
me.
sorry.
Okay, sorry.
It would, just to kind of be, okay, and Dr. Malik would too. Okay. All right, great. So maybe Richard or somebody on your team can maybe give some examples.
Add like one layer of like additional granularity to that, because Mina, I think, you know, case studies can be so helpful. There's a couple of layers of complexity for AP in particular that we've talked through, and that no individual recommended drug candidate is kind of recommended for all AP heroes. So there's kind of that piece. But the other thing that I wanted to touch on that I think Laura's question reflected is, you know, we have early evidence of accelerated aging in our AP heroes. And so the question of which controls are optimal controls and what might be masked in terms of aging by using a parental control exclusively, you know, I would love to kind of chat about. I don't know if that's something that has been a challenge in other contexts in which you've worked, or if there are opportunities to use, you know, other control data to supplement the parental control. But, you know, maybe just kind of weaving these pieces in, if that's not too much of an ask.
Yeah, so maybe let me just run through these two examples that we can talk about publicly. And there's a few more actually that we're working on getting both a case report, a couple case reports together, and some other videos and other information. So two completely different studies. One on the left was a family with a boy with Rett syndrome, and boys tend to not do quite as well. And they found us because we were working on Rett syndrome. We already had mouse data in male mice, you know, showing that a drug that we're working on, verinostat, was actually quite effective. And so they reached out and said, well, we don't know, you know, we never worked with males with Rett. We don't know, but we can use our nasal swab RNA-seq biomass approach, and let's see, right? So we did that, and verinostat was one of the absolute top drugs for this boy as well. And we've routinely seen it for female patient samples, but we had no idea what to expect for the boy. And so the clinicians started dosing him because this boy really was not doing well. And they saw improved sleep quality within a month, normalized blood metrics, so cortisol levels, some white blood cell counts, and other aspects were normalized, really for the first time ever in this 3-year-old boy's life. And then regained actually pretty substantial motor and cognitive function within about 3 months. And so it's been about 16 months now. He's still on it, still on the drug. It's actually bought them quite a bit of time to address some of the immune function, or immune dysfunction, I would say, that this boy has. It's you know, most likely caused by Rett syndrome, but it's extremely severe on the immune side. The drug did not help with that, but it helped with all these other aspects of both fine and gross motor control, and even the physical therapist that engaged with the patient on a regular basis really you know, wrote this whole letter looking at describing kind of what he saw and the improvements. On the right is a very different patient. So it's KMT2B dystonia. Was starting to have some dystonia, but really was extremely neurodevelopmentally delayed, nonverbal, minimal receptive speech even. And we found an existing drug, so it was a dopamine blocker, just anonymized here as Ravel68. And they actually prescribed it immediately, actually even before we had a chance to discuss the results. They saw very, very rapid improvements in sleep, and this patient was sleeping maybe 30, 60 minutes a night. It was really, really terrible. Could understand quite a lot of what his parents were saying within a couple of weeks. So for example, going to the store, like there's a video of him that you know, we actually have on YouTube now, where the mom basically tells me he's turned away from his mom and basically like, hey, can you hold on to the cart? And he just reaches over and holds onto the cart, which he wasn't able to do for a couple of years. And then actually after about 4 months started to communicate through sign language. This boy had to be taken off this drug because it's a dopamine blocker, and he started getting the extrapyramidal symptoms that so many recipients of these drugs get. And so actually we found a potential alternative now that we're going back to that team. And so hopefully this gives you, you know, an idea about how this has been working. We have now over a dozen patients that have all benefited from various treatments. There's a patient that started walking after 18 months post-status epilepticus where she lost the ability to walk. And about 7 weeks of dosing, she could walk independently. We also work on long COVID, we work on other disorders as well. But hopefully this gives you a sense of kind of the breadth of disorders that we work on and some of the case studies that we have here. The one on the right and another one, it's NF1X Mullen syndrome. Those are being written up for by the clinicians. Again, we don't control the clinical data, right? So it's not our story to tell from a publication standpoint. And then there's the other, just stop sharing here so you can see people. The other question around the age, we have not seen the age be as critical. So sex makes a huge difference, and probably not surprising. Age matters less. That said, if there's a very specific reason to look at that, we can. The big limitation of this study is it was only patients who were at the conference, right? So there are two biases. One is on the control side, whoever came with that patient, that's who was the control. So we talked with a number of families that had same-sex siblings who were unaffected. They may have been a better control, but they weren't at the conference, right? So there's a limiting factor there from a convenience standpoint. But the idea was let's start with the 20 or so patients, and then we can always do a follow-up study. So that's one part. I think that's sort of the biggest limitation. The other biggest limitation is in terms of the data, these are patients who could travel, where the families were who felt comfortable enough to travel, right? So this might be on the less severe side, with just patients who may have had fewer immune problems and other aspects that would have prevented travel. So there is a bias to the data. So I don't know if age really factors in that high. In that list, like would it counteract some of that bias? Probably not, is my guess. But we can always do a study to ask that. But that, of course, would then select for families where they do have a sibling, right? So they can't be a parent, if you want to go similar in age.
So you showed us examples of some positives. Have you had it where you had something that was strong and somebody tried the drug and they didn't see any benefit? And can you feed that back into your system to help try to figure out why it didn't work? Or like take samples from the patient after they've taken the drug and show that it didn't correct things, or it did correct things, but it's not helping clinically?
Yeah, so we have seen, the only thing we've seen in that case is temporary benefit. We haven't had a patient not benefit from the top predictions. But it may be like time limited, so maybe for 2-3 months. And we have resampled the patients at various time points. And in some cases, it depends on the family's participation to make sure that's possible. But usually we have that data. So far, we have seen consistency in those predictions. Where we start seeing, for example, with some of the patients where it works for a certain amount of time, then stops, usually that coincides with additional medications they're put on or off. And now it's a different state of that patient. And so it's not too surprising that it's going to be different. Again, that doesn't, there are other patients where it doesn't affect anything, but we think that in those cases there's something changed, and therefore now the data from before aren't valid anymore. But we haven't actually had a case where patients are on something, and again, we don't have it's not a controlled study, right? So it's not like we have that really clear data like you would in a trial. And there may very well be an effect where maybe families did try something and just don't tell us. Like that is a possibility that we can't force them to tell us, right? And so, because it's not a trial. And so it's a possibility, but anytime we've heard something back, it's been quite positive, or the sort of temporary positive. We have heard some negatives, but for example, that was actually with the KMT2B story. Somehow that the name of that drug got out, and a family tested it on their kid, And they didn't have any improvement. In fact, may have had some adverse events, fairly mild, and then they resolved while removing the drug. Well, turns out, and again, this was a personalized study, so we make, who knows what's going to happen with other patients. Turns out that patient also had leukodystrophy in fairly late stages, and also had KMT2B, and that's a completely different scenario. So to us, it's not at all surprising, and we actually work with numerous cases where there's sort of one mutation and there's the same mutation plus a different gene being mutated. We routinely see very different drug predictions. And so that's the challenge of taking personalized predictions and extrapolating to other patients, because those may not. In the population study, the challenge, and this is the biggest challenge, is figuring out, okay, what would translate to the most number of patients? And as we see in AP, which is not at all unusual, by the way, you may not have one drug that can do that. And that's, most clinical trials agree with that. Like you do this clinical trial, you're gonna get non-responders, almost always. Like they're very, like 2 trials ever, I think they've had 100% response rate. And so this is clinical practice, right? I mean, people are different. And so the hard part is figuring out then how you stratify them. We can do it computationally, but does it correlate with something more obvious, right? Whether it's the chromosomal or like the genomic aspects, or a phenotype, or some marker. And then we're always focused on, like, can we figure out a marker that can be used further? And so it looks like from the gene expression data, there may be something actually with AP where we get basically 2 groups based on gene expression that correlate with subgroups on the drug side. But I'm not surprised that we have multiple groups, and I would be very surprised and skeptical if we had only one here.
It's too bad you don't get samples after they're on the drug. It'd be really interesting to see how much you change it back to the norm, towards normal. What kind of effect you're having. Especially if they have minimal effect, could you increase the dose and move it more towards normal and get better effect, those kinds of things. But I understand the position you're in, but.
Yeah, and that's actually what we did with the boy with Rett syndrome. So especially with the verinostat, the clinician was extremely careful about dosing. And so she actually started at about 1/8 of the lowest approved dose. And then was slowly escalating it. And they saw effects within the first month, and it wasn't until, but in terms of the bigger effects, it wasn't until they got to a slightly more clinically relevant dose, right? So 1/8 of the lowest dose is nothing. But they were looking for side effects first, right? Just do no harm, and then maybe you see some positive effects. If it's not doing any harm, great, now we can maybe go a little bit higher and try again. And so one question they had was, well, should we keep going higher? And so it was after the first really early escalations that they were assuming they wouldn't see any benefits, but at least no harm. And we did see that the drug was still positive. I think it was like 67%, so still net positive, but lower from like the 5% rank to lower, which we expect. Every time we've seen a patient take a drug, it always decreases. And sometimes it's like just that one drug, even though there's a whole class of drugs that do something very similar. And then they kept going higher after that because it was saying, hey, it's still adding more could still help a little bit, right? But I think to your point earlier, now it was scoring less strongly. Like it's not going to have a humongous effect at this point, but more could still help more. And it did. So now they're at a I would say moderate dose of verinostat. Still not oncology levels, because again, you're not treating cancer here, but not the 1/8 of the lowest dose.
Mm-hmm.
So we have used this from a dose optimization standpoint. Excuse
me. Are we already coming up on time? My gosh, it goes so quickly. Are there any other questions from our audience here?
Maybe I'll ask a question if there aren't any. Where do you guys see, in terms of models or next steps, obviously we can share the drug list. That's not a problem. We don't like to do that in a presentation. There are many molecules that are could go straight into the clinic. All of them could go into a model. They're all purchasable. What would be the next steps? I mean, I know we kind of discussed that at the conference last year, but now that we have something, right? Any thoughts that come up?
Yeah, oh, I'm so happy that you asked this question, and yeah, we're so eager. So we have a researcher that's working with us that's stratifying the list, and so doing this in many different ways. So there are some candidates that were withdrawn or don't have safety information associated with them or aren't purchasable for some reason. So we've removed those, and then of course, and then we integrated safety data from like a variety of databases as well as pediatric safety data, and overlaid onto it the usages that are already known, that overlay with symptoms that have been prioritized by our community. And then on top of this, we have medication and supplement data from our community of what they actually use. So there's a lot that can be gained, both from understanding, the efficacy with which they rate medications that overlap with what's been recommended, as well as dosages that they take. And then we have a relationship with a pharmacology institute that we're working with to try and understand how we can approach dosage studies, for which we would be looking to evaluate dosage. And then we have some in vitro models. So right now we're in the process of evaluating a range of in vitro models to understand which would be best for 8P, and of course, which would be best is dependent on what you mean by what you're trying to model about 8P. So we have neuroprogenitor cells and fibroblasts and quite a range of cell types that we're working with to understand which ones are going to be best for evaluating the compounds that we feel are most meaningful. And then we have our clinical team that we're working with to understand the next steps that we can evaluate in the clinic to see if the pathways that have been modified that your research has led us to highlight, and whether or not we can have additional clinical validation that these pathways are indeed modified. And then one of the things that we think a lot about is traditional clustering approaches require a discovery and validation set. So we see this very much as the discovery set, and then we're looking to validate this as well, and just kind of understand how we can best do that with the data that we have stored and what prospective data that we need to collect. Yeah, and so then from there, we're able to screen the short list of drugs in a cell painting assay that we have going, which we're really excited about. That'll be at the end of this year once we've understood how to best stratify those hits and whether or not we need to do this, what subgrouping that we would need, to move forward with this, and how we could then extrapolate that subgrouping to the greater cohort. So lots of really good questions, and lots of tremendous opportunities that we're so grateful for our network of researchers and clinicians and families that are helping guide the way for this. It's a tough question, but it's an important one, and we're really excited about all of this. Well, I think I've rambled us over time. I'll just thank the Unravel team one more time, and thank everyone for joining. And certainly if there are any questions that come to mind for us or Richard and team, pass them along, and we're more than happy to help facilitate. And that goes for those who are watching the recording as well, who aren't able to join us live today, because I know there were a few that the schedules just didn't work. But yes, thank you again, and hoping everyone has a great rest of their week.
Thanks so much.
Bye-bye.